Hi there. Have you ever wondered how targeted sequencing readouts can help to improve NGS applications? Let's discover the advantages of targeted sequencing together. Conventional single-cell RNA sequencing amplifies all RNAs in a cell. This so-called whole transcriptome amplification workflow is powerful because it is unbiased, but it suffers from a shortcoming. In any given experiment, such as the one shown here, few genes vary between cells, and the great majority of mRNAs remains unchanged and thus do not provide any information content. Nevertheless, these non-informative genes consume most of the NGS reads in the experiment. Consequently, one would like to zoom in on the mRNAs circled in blue and specifically focus on these. How can this be achieved? Targeted sequencing focuses on specific mRNAs of interest rather than sequencing all mRNAs. To this end, selected transcripts are amplified using a multiplex PCR in which every mRNA target is amplified by a specific primer. Targeted sequencing has two major advantages over the conventional whole transcriptome amplification approach. First, it allows sequencing at a depth of 10,000 reads per cell versus 50,000 reads per cell in whole transcriptome sequencing. This leads to a five-fold reduction in NGS cost. And secondly, the dynamic range of the experiment is increased for the mRNAs in question. Let's look at an experiment in which Myllia systematically compared the performance of whole transcriptome sequencing to targeted sequencing. Here, scientists at Myllia selected mRNAs expressed at different abundance levels, so-called tiers. Tier 7 genes are highly expressed, Tier 1 genes are barely expressed at all. Then they designed a multiplex PCR panel allowing the amplification of 245 mRNAs, so 35 mRNAs per tier. Targeted samples were sequenced at 10,000 reads per cell, whereas whole transcriptome samples were sequenced at 50,000 reads per cell. The plot shows that, for the great majority of the mRNAs selected here, targeted sequencing is more sensitive than whole transcriptome sequencing. It also illustrates that even lowly expressed genes, which are often missed by whole transcriptome sequencing, can be captured using targeted sequencing. Overall, this data suggests that targeted sequencing is very powerful and captures transcripts at great sensitivity. Does this sound interesting to you? Well then, reach out to Myllia and have a look at our website in case you'd like to discuss your next CRISPR screening project, including a targeted sequencing readout.